Hi, I'm Thomas Maples from the lab of Dr. Adrian Rothenflew in the Department of Psychiatry at UT Southwestern Medical Center in Dallas. Today I will be demonstrating a simple procedure for determining the sensitivity of flies to ethanol-induced sedation. This assay is based on flies' loss of riding reflex and walking ability after exposure to ethanol. This measure can be used to determine alcohol sensitivity differences between different strains of Drosophila and mutants. We can also use this assay to induce tolerance in flies, tolerance being a reduced response to alcohol after repeat exposure. Let's get started. For this experiment, you will need the following: vials with 8 fruit flies apiece that you have collected 1 or 2 days before. You can do the exposures with either males or females, however, each vial should contain only one sex. An empty food vial that does not contain flies, a razor, 2 vial plugs made of a cotton-like material. If you do not have these kind of buzz plugs, you can also use cotton balls. A 1000 microliter micropipette with tips, 200 proof ethanol that contains food dye you can get at any grocery store, and a timer. The first thing we need to do is make an exposure chamber in which we will expose the flies to ethanol and monitor their mobility. To create this chamber, first cut a vial plug in half using a razor blade. Then push one of the halves to the bottom of a vial.